You need to access the data entry screen to enter results of the current sample and submit them to Bio-Rad. You can also use the data entry screen of eQuas Online to edit existing results before or after submission to Bio-Rad. To access the data entry screen, click the Data Entry menu. Alternatively, you can select the appropriate program and sample from the home page. Results need to be submitted by 2000 GMT on the sample due date. The sample due dates are printed on the specimen bottles and Bio-Rad calendar. On the data entry screen, select the appropriate lab ID from the lab ID list. Select a program from the program name list. Notice that only the current cycle of the program is available for each program. When a program is selected, tabs representing all samples in the current cycle are displayed. By default, the current sample, which is indicated using a black outline on the tab, is selected. You can also select a different active sample by clicking it. The page displays the analytes for the selected sample. You can enter results by clicking the Result field and entering the values. You can move to subsequent fields by pressing the keyboard Enter or Tab keys. Sometimes you might not be able to report the result for some analytes, such as when there are issues with the sample. You can then select the corresponding UR checkbox on the page. This displays a list in the Result field. Select the description that best describes the issue. Notice that as soon as you move to the next field after entering results, the submission data and time are automatically added in the row along with the user name. Finally, click the Save button to save the changes. The color of the Sample tab changes to indicate the current status of the sample. This is in accordance with the color coding used for samples in eQuas Online. Similarly, any late or amended results are indicated using appropriate symbols appearing next to the Result field and on the Sample tab after saving. All saved data is automatically submitted to Bio-Rad. In some cases, you might need to edit the results that were already entered for a sample. You do this on the Data Entry page, which can be accessed by clicking the Data Entry menu from the home page. Alternatively, select the appropriate sample from the required program on the home page. On the Data Entry screen, select the appropriate lab ID from the lab ID list. Select the program for which you want to edit results from the program name list. By default, the current sample is selected and its analytes are listed. Select the appropriate sample. Click the field you want to edit and make the change. To delete a result, select the result in the field and press the Delete or Backspace key on the keyboard. On the other hand, you can select the UR checkbox to report unreportable results. Finally, click the Save button to save the changes made. The color of the Sample tab changes appropriately, and, if appropriate, late results or amended results symbols are displayed in the Sample tab and adjacent to the specific result.